Okay, before we get into the next section, 9.2, I just wanted to talk a little bit more about the classification of lipids. As I mentioned, there are a few other topics. I'll go through this quickly. I left off with sterols in the last lecture, and so there are many uses for sterols besides steroids and cholesterol in our body. Such as bile salts, and we also use it to make vitamins. And so you've got this molecule here, 7-dehydrocholesterol, and when it's exposed to ultraviolet radiation, sunlight, it will actually undergo a pericyclic ring reaction and open up, undergo some rearrangements there, and will end up as vitamin D3. There's some later modification. It gets hydroxylated in the liver and the kidneys to get to the active form. And it helps promote the intestinal absorption of calcium. So if you don't have enough vitamin D, you tend to have weak bones. So this is a common additive to milk, because then you're getting the vitamin that helps support the uptake of calcium from the milk, and you get strong bones and teeth. There's a whole nother class of lipids known as the isoprenoids. These are derived from the isoprene unit, which you can see here. It is a conjugated diene with a methyl off of there. And it's amazing what type of molecules plants can construct from this isoprene unit. We also use it, we attach it to coenzyme Q to make ubiquinone. These are found throughout the body, hence the name ubiquinone, coming from ubiquitous. But like I mentioned, plants do some incredible things with this isoprene unit, forming these what are known as terpenoids. And here's one of them, which is actually found from sweet wormwood and is a new drug to treat malaria. And it's a really interesting molecule. You've got, this peroxide linkage here. But terpenoids are found in all sorts of interesting molecules. So these are plant-derived vitamins that are important to animals. And so a few of these are vitamin A and vitamin K and vitamin E, and you can see their uses there. And so again, these are all derived from the isoprene units. And bacteria actually use them to construct their membranes. So as we said, in eukaryotes, you don't get branching, but in bacteria, you may see it, and that is when they use these isoprene units to construct the fatty acid, not the fatty acid, but the chain here. It's not a fatty acid because if you notice here, it's actually an ether linkage and not an ester linkage. So the hydrophobic part is this branched component, and it's connected by ethers. And these are often used by extremophiles, meaning bacteria that live in extreme heat, where they need their membranes to be very stable. In fact, in some cases, you actually see the membrane where these units connect from one side of the membrane, which again we'll talk about just shortly, to the other side of the membrane, and actually kind of form a cyclic structure. These tend to be very stable. They have an ether linkage, which is much more stable and less hydrolytically labile like an ester. So you can see that over here where you get kind of these rings here, span the membrane, and those just end up being much more stable. You get a lot less flexibility in extreme environments. Now we get to my favorite molecules, which are the eicosanoids. So these are derived from the 20-carbon fatty acid, arachidonic acid, which you can see up here. This is often found in the second position on glycerophospholipids. And so if you are introduced to a phospholipase A2, this is actually released. And you have enzymes, specifically cyclooxygenases and prostaglandin synthases, that will take this molecule and wrap it up into all sorts of interesting molecules. And these molecules act locally. So these tend not to be transported throughout the body, but act in very close approximation to where that they're made. And so these include prostacyclins, prostaglandins, thromboxanes, lipoxins, and leukotrienes. And I wish I could just teach you a whole class on these things, because they're very interesting, involved in blood pressure regulation, blood clotting, allergic response, digestive activity, labor onset, inflammation, clotting, fever, pain, sleep-wake cycles. And you may be asking, why are they called prostaglandins? And one reason is because they were thought to be derived from the prostate gland. So if you need to go look that up, I'll let you look that up. But essentially, the prostate gland releases nutrients for sperm and other items in semen. And what they found out was when they put semen, which is composed of prostate fluid and sperm, on uterine muscle, so they excised some uterine muscle, I believe, from sheep and put semen on it, they noticed that the uterine muscle would start contracting. So they eventually identified the molecule that actually caused the contraction, and they were named prostaglandins. So prostaglandins serve in both ends of getting pregnant and delivering a baby. They're introduced into the vagina and cause contraction of the smooth muscles, helping propel the sperm towards the egg. And then they're also used to cause uterine contractions. So if you ever go get a induced labor, one of the first things they'll do is add some prostaglandin, I believe on the cervix or somewhere in there, and it starts causing contractions. And then they later introduce oxytocin, which also causes uterine contraction. But prostaglandins, really interesting. It causes smooth muscle contraction. And like I said, helps propel sperm to impregnate the egg, and can also be used to help expel the child when the time comes, as well as all these other things. Very, very interesting molecules, again, that act locally. So if you're stung by a bee, you're injected with the phospholipase A2, it can release the arachidonic acid and produce these prostaglandins. And that is one of the molecules that causes the local agitation and feeling of pain and heat at the site of injury. So the arachidonic acid can actually be used to make other molecules. And some of these other molecules are known as leukotrienes. These actually cause inflammation. And so if you've ever taken Singulair, it actually inhibits the synthesis of leukotrienes and helps with asthma and other anti-inflammatory issues. So these are present in small amounts in all body tissues and fluids. And like I said, it really depends on where in the body you're at and the function of the molecule. One interesting thing is, I'll go back, aspirin actually inhibits the synthesis. So if you're feeling pain or heat or a fever, it actually inhibits the synthesis of these prostaglandins and will alleviate some of those feelings of pain and heat. Okay, here's just some information on how they classify the prostaglandins if you wanted to know more about them. And if we go on, that is the end of lipid classification. Okay, so we're going to go into the next section, which is 9.2, lipid bilayers. This is fairly short, so hopefully I can get through this quickly. We're going to talk about how amphiphils can form bilayers and about the bilayer and its fluid structure and how the lipids can actually move. So lipids tend to aggregate together. Again, the reason for that is a hydrophobic effect, but really when we say hydrophobic effect, what we're trying to do is maximize the entropy of water. We've talked about this several times, but water, when it surrounds a hydrophobic molecule, doesn't like hydrophobic molecules, and so it adopts a very organized structure, kind of ice-like, around the hydrophobic portion. And that limits its mobility, adding order. And so to give back the mobility or the freedom of movement to the water, the hydrophobic groups tend to interact with one another. There is a little energy payoff there. There is some attraction there. But really what's driving this is the entropy. And so if you have these lipids, they tend to aggregate and exclude water. And so if you have these polar head groups, those polar head groups are going to be found towards the water, and the hydrophobic parts are going to self-associate. So what are formed are known as micelles, and these are formed by amphiphatic molecules, meaning those that had hydrophobic portions and hydrophilic portions. So such as soaps and detergents, and there's an example up here of one of those. And so if we look at these, they've got the polar head group and kind of this, what the diagram shows below, a van der Waals envelope. So it's kind of like a spike. And so these tend to form spherical compounds. They like to be very close and exclude water. If you make them much larger, then you get this interior water that is now surrounded by a very hydrophobic region, and that's unfavorable. And in fact, if you start getting water in there, then these may elongate. That separates these non-polar tails, allows water to get in there. And again, those are unfavorable. So when you get micelles, you tend to want to keep the greasy things in here and not include water. Well, if we change this amphiphilic molecule to give it two tails, it ends up being more block-like instead of more ice cream cone-like. And so with it being more block-like, it will still assemble into these micelle type molecules. But you notice as it elongates, you don't get the gaps where the water, so you can actually get kind of a more elongated structure here. And so we use this shorthand notation. So like a two-legged man with no arms. In fact, what would you call a man with no arms and no legs hanging on the wall? Art. What if he's in your front door? Matt. What if you're dragging him behind a speedboat? Skip. What if he lets go of the rope and he's just floating in the water? Bob. Okay, fun. So you see this no armed man right here. So just two legs and a head, and that is representing these glycerol phospholipids, or the other type, the sphingomyelins, or the other type of lipids where they've got a polar head group and these non-polar tails there. So that's what these little cartoons represent. And so what happens with these molecules is you actually get, instead of micelles, you get liposomes. So if you take phospholipids and you put them in water, they will self-assemble into a bilayer. And like I said, a lot of this has to do with having two tails and having this block-like structure. And with that block-like structure, you tend to get elongated structures, and you can actually have water on the interior because you have this hydrophilic or water-loving site as well. And so you can actually get these to be a fairly defined size. And again, you don't have a penalty really when you put water inside because these hydrophilic head groups are associating. So we call these vesicles or liposomes. And so you can actually, like I said, just form these, you mix them in with water and you agitate them. and you'll get these vesicles. And this was thought how life started to compartmentalize reactions was by lipids. You'd get concentration of reagents and reactants within here, and you could start to control those reactions. And now we still do that. We just refer to it as cells. So a membrane actually consists of two leaflets. So they're running in the same direction, and the legs, or these hydrophobic tails, are pointing inwards. So it makes it look like it's flat here, but we'll see that you can add curvature to these. And so with these lipids, the phospholipids and the sphingolipids, you can end up getting these bilayers, where the detergents or just fatty acid chains tend to form the micelles. And again, it makes sense because you've got water on the outside and on the inside, and then you've got the hydrophobic portions there. So the fatty acid tails are packed inside. And so you can look at these lipids here. I said that they were kind of block structure, but they probably have more of a spherical structure, and they fit together nicely to form these bilayers. Now, something that we need to understand about these bilayers is they're actually fairly fluid. So we're looking at the edge on of the bilayer. So imagine that we're just looking right down here. So all we see is the edge of the bilayer, and essentially just one cut of the lipids there. So as we look edge on at the bilayer, what we can start to understand, and we'll see some experiments later on, is that lateral diffusion, meaning a lipid moving in the same leaflet of the bilayer, is fairly rapid. In fact, it can move about 1 micrometer in a second, so about across the length of a bacteria. So from one end to the other end of the bacteria in about 1 second. So it's about equivalent, or similar to, I guess 1 order of magnitude slower than a small molecule traveling in water. Sorry, I said 1 order of magnitude, 1,000 order of magnitude. So it's not as fast as a molecule just traveling in water, so there is some resistance, but in reality, it's fairly quick, that movement. Now, if I wanted to go across what's referred to as transverse diffusion, or a flip-flop, that is rare and tends to be very slow. And the reason for that is if I did it, that polar head group would have to be buried within the interior of the hydrophobic portion. And so there's a big energy penalty to take something that is polar, put it into the interior of this non-polar region, and try to get it to come out the other side. So that's very slow. So hold that in the back of your mind, and we'll see some experiments and some other interesting features of real biological membranes that depend on this lateral diffusion and this slow transverse diffusion. So the big take-home message is these are not like 2 pieces of bread that make a sandwich. There is a lot of motion. Not only can the lipids move fast within a leaflet, these tails are wiggling quite a bit. So there's lots of wrapping and what they refer to as interdigitation, where these chains kind of insert themselves in between other chains. So these are fairly fluid. So they tend to be less fluid as you approach the polar head groups, but these tails down here are wiggling quite a bit. So about the viscosity of a light machine oil. If you're wondering what that is, that's like the all-purpose oil that, so not motor oil, but if you've ever used an oil that's clear in color, it's slippery, but not really, really thick, actually less thick than vegetable oil. So again, these hydrophobic tails bend around each other. The hydrophobic tails are of different lengths and have different flexibility. Some have those double bonds in there and some don't. So there's a lot of disorder inside this membrane. I just want to go back and compare, kind of our idealized version. That looks very organized right there. That just looks like everyone's in place. They're like little soldiers standing. This one shows that there is some disorder, but you look at the head groups and it looks like the membrane's flat across there. But in reality, the surface of the membrane is actually going to be bumpy. A couple of different reasons for that, but one of them is, you've got different lengths of the hydrophobic chains here. So you can see that, you're going to have some fat regions and some skinny regions along the membrane, and those are going to be dynamic and change over time. So the phospholipids actually up and down, the polar head groups will start to move into the water. As you start to drag them out of the membrane, you're starting to expose more of the hydrophobic tails. That's unfavorable, so it's more favorable and they'll be pulled back into the membrane. And water can actually, kind of dip down in here. You can kind of see that there's some polar groups, the red atoms there that they could interact with. But the water really isn't going to drop and be able to pass through the membrane. So you may find water out in this region, but you're not going to find any water in this very interior region of the membrane there. Now, when I talk about the fluidity, I need to add an asterisk to that because the fluidity of a membrane actually depends on the temperature. And we saw that before, that as I change the temperature, these can melt and turn into oils. And it really depends on the type of the fatty acids there. If we have really saturated fatty acids, that's going to have a really high melting point. And so in the terms of membranes, we refer to this as the transition temperature, where I move from a fluid temperature where everyone is free. So we say that we're above the transition temperature, and we actually have a fluid system to below the transition temperature where things start to go into a solid-like state, or a liquid crystal, a very organized state. Now, if you think about your cell membranes, you wouldn't want them to be in a very organized state like this because they would be very rigid. In fact, I gave an example of having a soup that actually has those little fat globules at the top. And when the soup is hot, those tend to be fluid. And if you put that soup in the fridge and bring it out, you'll notice that those fat globules have actually solidified. And you can actually take a fork or a spoon and break those in half. So below transition temperatures, the membrane's actually going to get stiff and crack. So you imagine that you don't want your membranes to do that. So we control the composition of our membranes very precisely to maximize fluidity. We don't want too much fluidity. We do want some organization. We do want those tails to stay together, but we definitely don't want them to transition into a solid state. Something that I remember way back when I took biochemistry was that reindeers in their ankles and lower legs actually have a higher degree of unsaturated fatty acids in their membranes. And it's thought that the reason that they have those is because they're standing in snow, and you don't want those membranes to solidify. You want them to remain fluid. And so you got lots of fluidity in the regions that are exposed to cold areas, and then in the other parts of the body, you don't have as much. You've got greater heat producing organs that keep your membranes fluid. But essentially in their ankles and lower legs, their membranes have a higher abundance of unsaturated fatty acids. So one way that we actually modulate that transition temperature is by including cholesterol in there. So cholesterol is a fairly rigid molecule. It fits nicely in the membrane, and it actually inhibits the fluidity. And so by adding more cholesterol to your membrane, the membranes become less fluid. And so actually you can find a significant amount of cholesterol in your membranes. But anyway, cholesterol has a purpose besides making things taste good and giving you heart attacks, and that is, again, adjusting the range of the membranes. Or sorry, not the range, the fluidity or the transition temperature of the membranes. So throwing cholesterol in there actually gives you a greater temperature range that your membrane will stay fluid at. So that is lipid bilayers. And I'm just going to briefly talk about the inclusion of membrane proteins. In section 9.4, it gets really deep. I think I might exclude some of that information there. There's just a lot more than we need to know. But in 9.3, it's very general, talking about membrane proteins. And so there's different types of membrane proteins. You got what are referred to as the peripheral or the integral membrane proteins. But membrane protein content can vary. So typical for eukaryotes is around 50% of the membrane by mass is proteins. So when we start talking about membranes, you really can't just talk about lipids. You actually have to include the discussion of proteins because they make up a significant amount. And I mentioned this, I think, earlier, but myelinated membranes that insulate, they have fewer proteins in there. You need to take advantage of the insulating properties of lipids. But protein-rich inner membranes for the mitochondria, where you're doing lots of chemical reactions at the interface of these membranes, contain actually a very significant amount. In fact, they contain by mass more protein than they do lipids. And so these membranes proteins have all sorts of functions. They can act as catalysts, they can mediate nutrient and waste transport across the membrane, and also involved in signaling, all stuff that we'll talk about. Now in section 9.4, they introduced the fluid mosaic model, but I want to introduce that now. So it was proposed in 1972, and the thought was that in this fluid mosaic model, if you actually take that name, the membrane is going to be fluid and it's going to be a mosaic, meaning that it's not going to be organized. You're going to have lots of different parts. Just Google mosaic if you want to see some mosaics, and you'll understand what they are trying to convey with that name. And so, again, the lipids form a viscous, so they resist flow, but they're two-dimensional. And the proteins can be inserted and integrated into these bilayers, but they can be in a couple different ways. They can be on the periphery, on the surface, where they can actually be easily attached and removed. Some can be non-covalently attached, or some can be actually linked to membranes. We'll talk about all those. Or they can be integral, which I have a picture here. So here's a porin, and we'll talk about those a little bit later, but it's actually sitting right in the membrane. Porins allow things to transverse the membrane. If we could see this from the top, you'd actually see a hole in there. But what we've got colored here, you can see the polar groups are colored red and blue, and the non-polar are colored white. So you can see that it's got a band of hydrophobic residues here, and those interact very nicely with the lipid membrane. The polar head groups on the lipids and the polar groups on the amino acids associate with water, and then the non-polar tails associate with this band around this integral protein. So again, this fluid mosaic model means that within this membrane, we've got lots of stuff. And so those orange globules, those actually represent cholesterol. You can see the different ways that proteins can be incorporated. They can actually go across the membrane there. Those are the integral ones. They can be bound to the surface. On the surface, we have these lipids with carbohydrates. Sometimes you have a lipid that's hooked to a protein through a carbohydrate chain. And Yeah, like I said, you just got yeah, proteins that actually will have a tail that's hydrophobic, and that associates with the lipid there. So all sorts of different ways to associate proteins with the lipid bilayer. So let's just take a little bit closer look at a protein sequence here and realize that, you know, we can start to identify these transmembrane proteins, proteins that actually go across the membrane. And what they're going to have is a sequence of very hydrophobic residues, and those hydrophobic residues are going to be found on the interior. So this is the erythrocyte glycophorin A. We'll see it, I believe, a little bit later in the role that it plays. But you can see on the groups that are inside and outside the cells, you've got a lot more polar residues. Those are going to be the ones that are red and blue. The non-polar residues are going to be the brown and purplish ones here. Dark purple, I should say. The light purple are kind of those polar non-charged residues. A better way to look at that is, in a second we'll look at that, sorry. In general, there's a couple ways that the proteins can cross the membrane. They can do it either as an alpha helix, where those alpha helix can actually bundle, or you can actually form what's known as a beta barrel. I showed you a picture of the one that I worked with, which was alpha hemolysin, that had this beta barrel that actually formed a hole in membranes. Maybe we'll talk about that a little bit later, but here's a bunch of beta barrel proteins. These are, I believe, porins, bacterial porins that allow, again, the movement of goods in and out of bacterial cells. And you can classify them based on the number of beta strands that they have. You know, so you can get up to 26 strands to form a hole in a membrane. Again, those, it's kind of hard to see, but that light tan band is the region that the nonpolar tails would interact with the protein there. Okay, so for any of these, the regions that are embedded in the protein need to be hydrophobic. So just some notes about the arrangement of these proteins in the membranes. They tend to be asymmetric. So these integral membrane proteins tend to be inserted in a particular direction. So you get specific domains that face into or out of the cell. All membranes are partly exposed, at least of one side, so there aren't known proteins that are just buried on the interior. And that really wouldn't make sense if there were. I'm not saying that we'll never find that, but if a protein's just buried in the interior of a membrane, you know, you wonder what it's doing there. It's not going to have access to the outside or the inside of the cell. So if it was just on the interior, you know, it'd probably have some type of interesting function if we ever find those. But generally, they're going to be exposed on one side of the membrane because they need to interact with something in the aqueous environment. So you get glycosylation, and that tends to be much heavier on the extracellular domains. Those are that are on the outside of the cell. Okay, so like I said, you can look at proteins and start to figure out those that are found in membranes. And what you do is you plot their hydropathy index. And if you start to see big spikes in regions that are very hydrophobic, those could either be buried in the interior of a protein, or it may indicate that it is a region that is embedded within a protein, or sorry, a membrane. So like I said, if you see these, and we've seen some of these plots before, but if you see a large region, that may indicate that this is a transmembrane. The fact that you got some here that like to be exposed to water, you got some hydrophobic, and then you got those that like to be exposed to water, that's a good indication that this may be a transmembrane protein, where you've got one end that's on, let's say, the outside, one end that's on the inside, and you have to transverse that bilayer. Sometimes you see this case where you got very hydrophobic, and then kind of, you know, drop down in our hydropathy, and then hydrophobic, and maybe even a little bit of hydrophilic, hydrophobic. And if you see this repeating pattern, that's probably a good idea that you've got these transmembrane helical proteins. So in fact, each one of these is color coded. So that red region correlates to that. This orange, they didn't match up the color very well there, but the yellow region here, the dark green, the light blue, and the dark blue, and then they screwed up. So yeah, they messed up on the color. In fact, it goes from yellow to dark green, or sorry, light green to dark green. So yeah, I thought they're supposed to match up. So send that to the authors that there's a mistake. So this is bacteria rhodopsin protein, and that is its plot. So when I first looked at that, I thought, oh, that's so nice. It all matches up. But yeah, the colors don't quite match up. They don't have purple. They missed purple. Okay, so let's talk about one of these transmembrane proteins, specifically a beta-baryl. Like I said, these examples are porins. So in porins, a beta-baryl with at least 16 strands forms a solvent-accessible central channel. So solvent can actually get down in here and actually move through it. Oftentimes there's some regulation residues in here that prevent that. So something needs to bind, and then that'll cause a conformation change and let other things go through. So in gram-negative bacteria, porins permit entry of small polar molecules. They're also found in the mitochondrial and chloroplast membranes of eukaryotes. So this is what's known as UMP-F right there. So again, what you'd expect is on the outside of this beta-baryl right there, you'd expect to find hydrophobic residues there. And if you remember with the beta sheets, they alternate. On one side, the side chain's pointing out. On the other side, the side chain is pointing in. So if you were looking at a porin in one of those plots that we just looked at, what you'd expect is kind of a alternating hydrophobic, hydrophilic, hydrophobic, hydrophilic amino acid sequence. Again, one's pointing out, and then one's going to be pointing to the interior of that beta sheet. So this is just a trimer of these porins. Here's a different view of it. So they may form trimers. So each one of these is just a porin here. And you can see that there is a solvent accessible region down the middle. So this is looking from the top down, and this is looking from the edge on. So carbons are colored in green except for the hydrophobic residues. I believe the tyrosine and the phenylalanines and tryptophans are colored in gray. And so you can see that the gray, you kind of form this hydrophobic belt around these aquaporins here. So this doesn't include any lipids. You just see lots of kind of green spots and, you know, less density of the blue and the red. And you get these hydrophobic residues that would be very happy to interact with the hydrophobic tails. Okay, so proteins in solution are to a great extent stabilized by exclusion of the hydrophobic residues from water. So we talked about that in folding, how we exclude those hydrophobic residues and put them on the interior. So what stabilizes an integral membrane protein, something that's stuck in the membrane? So the interior residues of these membrane proteins have hydrophobicities that are comparable to the interior residues of soluble proteins. So like I said, kind of the same type of effect. We want to bury those hydrophobic residues either on the interior of a protein, or if they're on the surface, they need to interact with lipids. So surface lipids, exposed residues are more hydrophobic than the interior on the protein. So the less hydrophobic interior is excluded from the lipid. Oh, as I'm reading that to you, that doesn't make sense, but that could be that it's a long day. But this is important right here, the low polarity of the anhydrous surroundings. So essentially what's surrounding those proteins likely strengthens the hydrogen bonds in the protein. And the reason for that is you don't have water on the interior of the lipid competing for those hydrogen bonds. And so that's likely to increase the stability and the importance of those hydrogen bonds in keeping those proteins folded. And you kind of can guess that when you look at these porins here, because remember that you get a hydrogen bonding between the backbone here. And so if you bury this in a hydrophobic environment, those hydrogen bonds are likely going to be more important, and you don't have the water competing to break those up. Okay, so proteins can be tagged with a hydrophobic residue that will actually allow them to interact with the lipid. So we've looked at the structure of the proteins and we've got these hydrophobic residues, but what if we put a TEL on the protein? I don't want to make you too sick, so let me see if I can find the example in the earlier slide. Here we go. So if you look here, we've actually got a lipid that is attached to a protein by a TEL. Or this one right here, we've got a protein that's attached to some type of hydrophobic residue, and that hydrophobic residue interacts in the lipid. So let's talk about a couple ways that that can be accomplished. So you can do some prenylation where you're actually adding these prenyl groups, and there's two types that we'll talk about: the farnesyl residue and the geranyl residue right here. And the type of residue that you attach to the protein depends on the sequence. So for prenylation, it generally happens at the C-terminus of the protein, and you have a sequence C, so we got a cysteine. Then you tend to have an aliphatic, meaning a non-polar amino acid, and then you tend to have Y, which determines if you're going to attach a farnesyl group or a geranyl. So if that Y is alanine or methionine or serine, then you tend to add this farnesyl residue. And the farnesyl residue is actually attached by a Michael reaction from the cysteine sulfur to that double bond. So if you look at this, this is very similar to Michael chemistry. So you can actually attack a double bond there and form a bond between the sulfur and this beta carbon right there. Now, if Y tends to be a leucine in this sequence right there, then you get the geranyllation occurring. And then what happens after you get this prenyl group, either the farnesyl or geranyl, then you actually cleave off the XXY sequence and methylate the C-terminus there. So this is found at the C-terminus. So you have those sequence of amino acids and you have the cysteine, that then you add this hydrophobic TEL. And this hydrophobic TEL can then interact and anchor that protein within the membrane. You can also acylate different residues. So you got myristylation. So you actually attach C14 saturated fatty acid to the N-terminus glycine here. So amide bonds tend to be fairly stable. So once you add this myristyl fatty acid, it tends to be stuck there for kind of the life of the protein. But you can get palmitylation from palmityl fatty acid, and that tends to be more specific for cysteine residues. So you get what's referred to as a thioester. And thioesters, actually we'll see later on, those are actually fairly labile. And so you can use this, you can put the palmityl group on and off, and so it can be used in signaling, so it's reversible. And again, what these little TELs do is it allows this to be embedded in the membrane, and so then you get these little TEL groups that are embedded in the membrane and then the protein that is attached to them. There's one other linkage, and that's these glycosyl phosphatidylinositol linked proteins, or GPI linked. They occur in all eukaryotes, and they're particularly abundant in some parasites. So they're located exclusively on the exterior surface of the plasma membrane, so on the outside. And so what you have is, here's your phosphatidylinositol. So if you look, you got your, it's a glycerol phospholipid, and you've attached the inositol sugar here. And that inositol, you actually have a tetrasaccharide linkage that then connects to another phosphate group, specifically phosphoethanolamine. And then from that nitrogen, you form a covalent attachment to the protein. And again, this is what would be embedded in, well, actually, this is what's embedded in the lipid, and this is the polar head group. So this allows that to the protein to be extended above the membrane. So I just want to go back and show you that original diagram, and you'll actually see where that type of attachment is represented. So that is this type of the GPI anchored protein. Notice that you've got this saccharide that's linking the protein to the lipid bilayer there. So that closes up this section, and so that ends 9.3. Like I said, 9.4, man, they just throw everything at the wall. So I'm going to try to streamline that and get that up tomorrow, but it won't be nearly as comprehensive as what they're presenting in there. They just throw a chapter's worth of topics into one section. But we'll get that up early in the morning, and then we will have one more section to talk about, and that is transport across membranes. So we should get that up hopefully I'll get that up on Thursday, so Friday can be a study day, and again, the exam will be on Monday.